Jaume Bonet, and he will speak about TopoBuilder, expanding the and functionalizing the protein fold space. I think it should be behind.
Oh yeah, there it is. Oh. Wasn't there a pointer? Oh, yeah, sorry. Yeah. Okay, perfect. Yes, so yeah, I'm going to be talking about the work that we've been doing in terms of creating de novo functional proteins. So I'm going to go a little bit over why we're doing this and show you some of the applications that we've developed with it. So okay, so as you know, the sequence of the protein defines their structural conformation. And in the last 10 years, the number of sequences that we are getting is increasing exponentially, thanks to the new transcription techniques. And also, one could see that every year we do get more and more crystallized structures. Regardless of all of this, the truth is that when we check on the new domains that we find, it happens that in the last 10 years, only 2 new topologies have been added to Cath. This kind of suggests that we are reaching the limit on the topologies that nature has actually explored. So the question that we have is, are we actually able to explore this space that nature has not? And of course, this is not questions that we are the first one to ask, but we are asking it from a more functional perspective. So, well, as you all know, vaccination is one of the most successful treatments on humanity, and still we are missing vaccines for some very important viruses like malaria or Zika or HIV. And in our lab, we have taken the challenge to actually kind of tackle this issue with an idea that is called reverse vaccinology. And this idea basically tells you that if we know from literature neutralizing epitopes that create neutralizing antibodies in humans, can we actually take these epitopes and create new proteins that are vaccine candidates for the viruses from which these epitopes come? And we try to answer this question over this virus, which is called RSV, respiratory syncytial virus, which is a virus that affects kids and the elderly especially, to try to see if we can actually solve this problem. So the idea is basically the same as functional motif transfer design, which is that once you identify the neutralizing epitope, you take it out of its original context, and you put it into a new scaffold that will both stabilize it and present it to the known antibodies properly. The problem that we are facing is that as the complexity of the epitope grows, it is more and more difficult to actually find structures that could fit that particular epitope and also present it to the antibody, to the known antibodies and to the unknown ones properly so that it will be also functional, so that we could clip the functionality. So what we thought is, okay, if nature is not providing us with the right carriers to put our scaffold, our motifs, let's just build them ourselves. And again, building de novo scaffolds, this is not something that we are inventing. This is something that it's been tackled in different ways. And most of the current methodologies to tackle this idea come always to the same, which is first define a particular topology, get a set of reliable backbones that belong to this topology, then through protein design, get sequences that would stabilize these backbones, and finally assess them computationally and then characterize them experimentally. And this has had a lot of success, both in creating both in de novo replicating known or existing topologies, and also to create topologies that are out of the natural space, which would be the case, for example, of top7, which you see here. How we see the problem is that these de novo topologies, in general, they tend to be made out of like a set of standardized rules, which make them, sure, highly stable, but also very idealized, or handcrafted. But these, I mean that they are created with the specific rules to define that particular topology, so not in a generalized way. And this basically is because this step, the part of defining the topology, is not trivial, and it's the bottleneck of this whole process. Just as a demonstration, I guess, if you go to the PDB and pick all the de novo proteins that are currently deposited there, and you separate them by the institutions who have made them, you could see that basically there are some that are making quite a bit, and then some people that can actually do some. When you go to even de novo structures that have the two main types of secondary structure, this is still even more obvious. So this means that apart from the tools that have been developed to actually create these topologies, there is also a level of expertise that it's inside the people that is there that limits the ability to actually build this kind of topologies. And this is the kind of thing that we actually wanted to skip. So we thought, how can we actually create these de novo proteins in a way that it's more accessible? And we stumbled upon this concept of the form. So a form is a way to reduce the way in which you define a protein. Basically, you take the protein and you divide it by layers. And then any given protein would have layers from A to Z, and each layer would either contain alpha helices or beta strands. If you do this, then the idea is that by just providing simple strings that define the layer and the number of secondary structures and type of the secondary structures, you can already create, well, only with these undirected architectures, which is the definition of the placement of the secondary structures of the topology you want to build. So if you want to even go a little bit further, you can create a small regex there, like the one that you can see there. So this one, for example, will actually define multiple types of architectures. And then when you start to see, because of course you have to connect the secondary structures to get a protein, when you start to see how each one of them can generate a great amount of topologies. So basically, although this definition is quite simple, potentially you can end up defining much more topologies than the ones that are currently classified in Cath. So, okay, the way that we try to exploit this, we actually developed this idea of the TopoBuilder, which will take you from the string definition up to a population of design decoys. Walking like step by step through it, basically what we do is we take the string definition and build the two-dimensional positioning of the secondary structures. And then through parametric construction, we build the idealized secondary structures that will be placed in these positions. Now we can join them as a graph, and through a graph walk, well, it should have moved, we can actually get all the possible configurations of these secondary structures that will produce at the end a connected protein. So once we have this, the next step is to try to bring back some natural features to these secondary structures. To do that, what we do is we submit each individual layer of our constructs to the protein database. And then, for example, for this case, we would find some protein just like this. And then we will pick corrections from the what we call the flat topology. So to show an example, the dark gray would be the distribution of the database for this topology on the angle that the betas create to their own beta sheet. If you get it flat, at the end you will generate a set of topologies in which the angle is still 0, which does not match the trend of the natural proteins. But if you correct it, you can actually mimic the trend of natural proteins. So once we have placed these secondary structures, from here we can take distance and angle constraints that we are going to use moving forward. The second step is tie these structures together. And to do that, we query on a database of loop classification. It's a database that classifies loops according to the geometries of their secondary structures. So you can imagine that we can only need to set up the two secondary structures and bring back all the loops that will match their particular paired disposition. From here, we bring the loop lengths, and we bring the phi-psi data from these super secondary structures. So now that we have this plus the constraints, what we do is we run a constraint ab initio protocol that it's called FunFolds that we added to Rosetta that allows us to fold back the structure according to the constraints that we have previously generated. We use funfolds instead of a regular constraint ab initio because we added the ability to have a functional motif enclosed and keep it as is during the folding process. The funfolds also, well, through Rosetta, we also include the protein, also the design of the residues. So this is how we are now doing this thing, how we are carrying out this idea, and I will show you a couple of examples of what we've done with it. Yeah. So as I told you, what we are trying to do is create vaccine candidates, and that our virus of our model virus is RSV. So in this case, we took the site 4 and site 0. So you can see that this is a two-piece motif, and this is a virus strand. And we created multiple topologies that we expected to be able to fix this motif and present them properly. In this case, we did fix this manually because we were aiming to create at the end topologies that on the surface of the context, so around the motif that we are fixing, would also mimic as good as possible the surface that surrounds them in the original protein. So they went through characterization and selection, and at the end we did find some designs, or particular designs that were actually, they were pretty stable, and they bound properly to the target antibodies. As a matter of fact, we did get crystals for both of them, and you can see that in both cases the designs are properly matched with the final crystal structure. So what we did with this very quickly is that we took these two plus a design for a third epitope that was from a previous work and shoot it into mice. And what you can see is that when you shoot the three of them, you actually get neutralizing titers for RSV infection. Also, what we did is we actually we used them to boost immunity over monkeys that had been previously exposed to RSV. And in this case, what is interesting is that we were actually able to reshape the behavior of the B cell population. And you can see on time, for example, for site 2, the B cell population that attacks this particular epitope lowers, but by boosting, this one is kept high. So this was one of the examples. Then, of course, we also tried to make more of these scaffolds for site 4. And what is interesting here is that all of these scaffolds actually have the same topology, so all the secondary structures are placed in the same conformation, but they are linked differently. So the loops, or on the order in which they are in the protein, they are different. So from several of them, at the end, we selected these three. This is actually the one that you've seen before, so these are the two news, that seem to fall properly in silico. Out of the three, so of course this one was binding okay, and we found this other one that also bound pretty good. This one, because it really doesn't seem to be working, we disregarded it. And again, for this second one, we again got a crystal that also showed that the design was properly mimicking the spec or the final structure. So I think this is relevant because being able to swap loops allows us to better mimic, the context in which the original epitope was found, but also because this has potential for loop functioning. Okay, and just a last example, and this we did in collaboration with Leo Scheller here in Basel. So he had developed these receptors that basically, in which he can basically change the affinity domains and the intercellular domains so that through different stimulus, they can respond differently. So what we wanted to do is actually create a bifunctional de novo structure. So actually carry two functions on the same de novo protein. And we did that, but including in the same protein, Mora on one side, sorry, I don't really see the, okay. Mora on one side, and 2D10, which is this other epitope, on the other side, back to back. The idea was to create a design that would bind both antibodies at the same time, which we checked by SPR, and you can see that you can provide Mora, and then when you provide 2D10, you are also binding both of them. Again, when we tried this in Leo's design, in which he changed the affinity domain so that it would be the Fabs of each one of these two antibodies, we can see that actually we're able to generate a signal on the cells that have this receptor when we provide the functional design, but not when we provide designs that only carry either one or the other function. So the good thing about this is that it's showing us that by fine-tuning control of the position of the secondary structures, we can actually decide and carry multiple functional motifs and decide how they are going to work with each other, either cooperatively or compete between them, etc. Yeah, so, well, with this, well, I hope I show you a little bit how TopoBuilder is working and how we are trying to tackle this idea of creating functional de novo proteins, that it has the potential to be a very systematic tool to explore the conformational space, and that by creating this simplified way of defining a topology from the start, we are kind of expecting to be able to democratize this idea of de novo scaffolding. And yeah, with that, I want to thank all our collaborators, the people from my lab. Xander has been helping me on the benchmarking of the protocol, and Fabian and Che are the ones that have been spearheading all the biological applications, and all of you for your attention.
So questions?
Have you tried doing the same pipeline without the step where you take the helices and then you fold them a bit to that they look like natural instead of
to avoid the naturalization, you mean?
Yeah.
Yes. So the pipeline, actually, we develop it in a way that it's kind of a plugin. So every step is, well, not every step is optional, of course, but some of the steps are optional, and also some steps can be solved in different ways, as long as, you know, at the end of a particular step, the kind of output is something that the next step can read. Which makes sense. Yes, so sorry, well, I should have gone the other way. Yes, so in this case, for example, we actually didn't apply this. So to these designs, you see that we are here literally just marking this. So what was done here is that while they were building these topologies, they had, like you can see here, the surface of the protein. And they were building topologies in a way that they were making sure that the secondary structures will not go out of the surface. You know, you build it, and I mean, you look at them and you just see it. So you can, yeah. So we've also built them without applying this step.
But in most cases, you have more success while applying it.
When you're not using a functional motif, it does perform better applying it. When you're using a functional motif, it depends on the motif.
And last question, do you have like a part of code or something that helps to do that? Let's say I have a barrel and I would like to make it more natural.
I mean, yes. So we do have it because in order to actually place the functional motifs, what we do is we take them. How can I explain it? So when we build the parametric structures, we build an edge for each structure, and then we build around it. When we take a functional motif, we build the edges inside these functional motifs so that, you know, we can bring them to our way. And the only thing is that then when we create back the atoms, these atoms come created by the motif instead of a parametric. So basically, we call it, well, so basically one could take a particular barrel, you know, it will, like you could tell it to consider it all of it a motif, it will decompose it, then you take out well, a secure thing that there is so the motifs don't move, and it would just do that. Yeah.
Thanks.
Yeah.
Okay, if there are no other questions, I have, from what I understand, I put in design. There's one group that did many designs. I mean, all know which group it is. And but one reason for the success, from what I understand, is that they actually not only do design on the final structure, they actually do folding simulations to get there. To avoid getting struck. But you don't need to do that. So
I think there are different ways to solve it. We do folding simulations. The difference is that, so when you cannot just do unconstrained folding simulations when you expect to build a topology from scratch, also because you don't have a sequence to start with, in a sense. I mean, when you see on the, generally you fall on balance and then from here. So you do need constraints to bring constraints back to the table. And the issue is how you bring those constraints. And you know, in other cases, it's been like top 7 was, I mean, you look at the paper, it's like manually made, but we cannot all do that. So we are kind of bringing these constraints from the parametric part, but we bring them also in a way that, you know, when we refold, that there is margin there for, and you see that, well, when I was showing this distribution on the angles, if we set something at 60 degrees, it doesn't come at 60 degrees, it comes around. So enough that it makes sense, you know.
Okay, then I think we thank all the speakers, and we'll be back here in 40 minutes. So.